the invitation, and thank you for joining. And I didn't realize I have the honor of starting the seminar series. So thank you again. So let me start by acknowledging the fact that a lot of this work has been funded by NSF, and I'm going to talk about a lot of work and say we, and it's really the royal we, right? It's been grad students, postdocs, technicians, and then colleagues that have collected a lot of this data over the years. Emily Bernhardt at Duke has kind of been my primary collaborator throughout this whole period. I still work with her. I started working with her as a postdoc and still working with her. Justin Wright and Ryan Emanuel, both from Duke, also have been very close collaborators throughout this process. And these are just some images of lab groups over the years. I guess I couldn't resist, and I think our approach to understanding salinity is similar to you guys' approach, but we come at it at slightly different angles, and I just couldn't resist using these images from the Olympics, right? To think about you guys' approach versus our approach, right? But in the end, we're all interested in very similar things, right? Trying to understand salinity and ocean water interactions, right? And I always like to start by telling people how I started doing this, because I did not seek this out, right? So when I started a postdoc at Duke in 2006, we were studying wetland restoration. And we saw these trees that were just like growing like gangbusters. They planted them in an old agricultural field. There was fertilizer legacy in those soils. The trees were growing like crazy. And we went around trying to find nearby forested wetlands that could serve as our controls for the restored site. And we found a couple, but there were a couple of those sites that we were studying that during the time we were doing this research, the trees were dying, marsh vegetation was coming in, the water was getting higher and higher. I always tell people one of the sites I started working at, I was wearing knee boots, then I had to wear hip waders, then I had to wear chest waders, then we just abandoned that site because it just got so wet. And so in some instances, the forest was turning into a marsh. In other instances, the forest was being swallowed by the water, and there was no marsh vegetation coming in. And so that got us really interested in, what are the drivers, what are the causes, what are the consequences? Like I said, we've been doing this work for a while, and a couple of years ago, this guy from Science Friday, the NPR show, came to do a video. Yeah, it's a radio show, but they also do videos, and they created this video called The Seeds of Ghost Forests. If you just type it into Google, it'll come up. It's really cool. He did a really good job. Ryan Emanuel and Emily Bernhardt are on there. They're amazing scientists. I love that he made me sound smart, and so I love that video. So I always like to mention that. So the Swissler was mentioned before. I know some of you are involved in this saltwater intrusion and sea level rise research coordination network. I just wanted to put the website on there again. It's just a way to try to coordinate and see, there's a lot of us studying salinity, a lot of us studying these changes. I think if we, combine forces and try to understand what we're doing, what works, what doesn't work, I think we'll get a lot farther, and we can get a lot more insights. This is we did a review about kind of the socioeconomic consequences of saltwater intrusion and sea level rise, and they had this really nice artwork, so I thought I want to put that on there. I give talks about ghost forests a lot, and people always want to see pictures of them, right? And I also like showing pictures because, this is what they look like here in North Carolina. They might look a little bit different. I know you guys are working in Chesapeake Bay. It might be different in other parts where other people are, but I always like to show these, and I like to remind people these are expanding, and I've found, in this time of a year, they also seem to be expanding here into Raleigh. These are pictures from my neighborhood. I feel I've talked about ghost forests so much that I'm affecting people's Halloween decorations. We know what, ultimate drivers of this, at least here in the Southeast, we know that sea level rise is accelerating, and this paper talked about measuring rates upwards of 10 millimeters per year compared to the global rate of 3.5 millimeters per year. So we know this is definitely happening, and it's having an impact in our coastal ecosystems. Um, this is just a map of North Carolina, showing projections. This is actually a little bit dated now, but it's still a good map, but showing areas that would be underwater with 1.1 meters of sea level rise by 2100. I've been doing a lot of work in this area, this Albemarle Pamlico Peninsula. It started by the restored site, which is this red dot right here. Um, and I'm like showing this map just to again show this is what it'll likely happen by 2100. When I give this talk to general audiences, I also make the point that these maps are really not the best way to express this information, right? And it makes me think of this cartoon, right? It's like terrible things happen more slowly. We need to find better ways to talk about sea level rise rather than showing these kinds of maps, right? If your house is right there, what does it mean that it's going to be underwater by 2100? How do you take that information? How do you make decisions? How do you act rather than get scared or just say, whatever, I'll be dead by then. I'd like to mention a paper that we published a couple of years ago where we tried to look for the literature as much as we could in terms of effects of salinization of coastal wetlands. We had colleagues from Europe, so we found examples there. We had colleagues from Australia, we found examples there. We, of course, found lots of examples in the US. I'm sure this is happening in South America. It's happening in Africa as well. We just, at the time, we didn't find evidence in the literature. There might be more evidence in the literature now, but we should go back and look. In terms of the spread of these ghost forests and the loss of mortality, right? Colleague Elliot White and others put out this paper in 2021, which I think is a really good analysis. They used remote sensing, and they came up that the loss between 1996 and 2016 has been 13,000 square kilometers, a higher loss rate than mangroves, right, which received a lot of attention. They tried to account for that variation, and the rates of sea level rise, the number of storms, and elevations explained a lot of the variation, the spatial variation, right? You can see here South Carolina and North Carolina are hotspots of this loss, but also Florida, Virginia, losing a lot of these forests, right? If we, sorry, well, if we focus here in North Carolina, get rid of this. There's been a couple of studies that have used slightly different methods. Again, this is focusing on this kind of Albemarle Pamlico Peninsula. Lindsay Smart did a study where she used LIDAR to estimate the number of ghost forests. She used LIDAR combined with field surveys to estimate the amount of carbon. So they estimate about 0.13 teragrams carbon lost in that time period. And again, 167 square kilometers of ghost forest in that time period. And again, DC has an area of about 171 square kilometers for frame of reference. Emily Urey did another study in pretty much the same area. She focused more on the Alligator River Wildlife Refuge, so it's a slightly smaller area. She used the Landsat record to categorize these, forest, shrub, ghost forest, marsh, or water, and mapped it out. You can see that the ghost forests kind of seem to come up and then disappear and then come up again. They seem to come up after periods of drought and hurricanes. So in 2011, 2008, 2009, and 2010, there had been a number of droughts, and then we had a hurricane. And so it seems to be that double whammy of various stressors coming in that lead to the spread of ghost forest. She found a similar amount of ghost forest creation in similar time period as Lindsay Smart. So we know that the effects of salinity vary across space and time. This figure is from that review that I mentioned earlier, the Herbert et al. 2015 paper. And I just want to, use it to, show that these effects will vary from days to weeks, months, years, and decades, and they'll affect both biogeochemical, community, ecosystem. And so what I'm going to show you is just some different data sets that we've done across these different both time scales and spatial scales, um, trying to disentangle the effects of salinity across all these different scales, right? Starting kind of a large spatial scale, again, in North Carolina, focusing on the Albemarle Pamlico Sound. We were interested in salinity as a very important driver, right? And we were trying to synthesize as much salinity data as we could find. This goes back to the 1960s. You can see that there's a gradual increase in salinity, but there's also pulses of salinity that are happening. A lot of those pulses happen during drought periods, but there are also some pulses that happen after storms. And the storms, it also depends on what the path of the storm was. Is it over land? Is it over water? If it's over land, usually you get fresh water that comes in and pushes out the salinity. If it's over the water, you get salinity pushed onto land. So here's what salinity used to be in the 70s, 2010s, and the salinity change. You see a lot more warming colors, places where we're having more salinity in this area. Abby Batachan did a study a couple of years ago where we try to understand the role of ditches, right? So this landscape has been ditched and drained for agriculture, and those ditches are very good at moving water off of the landscape, but that those ditches can also allow salinity into the landscape. And so this is what it looks like right now. We try to recreate what the waterways used to look like before the ditches were there. And then we calculate a saltwater intrusion vulnerability index. You can see that warmer colors suggest higher vulnerability thanks to the ditches facilitating the movement of salinity into these systems. So one of the first papers that I published related to this, I guess, is now almost 10 years old. Yeah, I guess over 10 years old, right? So it goes back to that restored site. We had found that in years that there was no saltwater intrusion, the site was a nitrogen sink. But in years that there was saltwater intrusion, the site was a nitrogen source. And this had to do mostly with ammonium. So this is just chloride on the x-axis and ammonium in the soil solution. We did field and a microcosm experiment. I'll talk about that more in a second. In surface water, we also saw ammonium going up. So this is the restored wetland. Remember, I mentioned that there was fertilizer in those soils, so ammonium was really high. We also saw ammonium going up in those control kind of mature wetlands nearby, but the ammonium was a lot lower than what we saw in the restored site. And we think this is because of legacy fertilizer in these sites. Now, this was happening when we had drought. Right, so is it the salinity? Is it drought? Is it the two of them? We couldn't really tell. So we designed a microcosm experiment where we took a lot of soil cores and we brought them back to the lab. We had a drought treatment, a flooded treatment, and then we wanted to look at salinity, but disentangle the effects of salinity, particularly the effects of sulfate. So we created artificial salinity with and without sulfate. And then we also added sulfate alone at the same kind of concentration as we added in the salinity. And then we had a control that was just DI water. And so you can see that the ammonium increased in the salinity treatments, whether or not we had sulfate. So what that told us is that it was the other cations in salinity, not the sulfate, that was leading to this increase in ammonium. And what we think was happening was basically we were doing a salt extraction from these soils, right? We were adding salinity and that was doing cation exchange, and the ammonium was becoming released and going into the soil solution. One of the surprises from this experiment was that we found that salinity increased methane. And so this was, completely opposite to what we had expected. There had been many experiments and meta-analysis and different site studies that have seen that as you increase salinity, methane fluxes go down because sulfate is more thermodynamically favorable. So sulfate reducers outcompete methanogens. And so methane usually goes down, but we found that the methane actually increased, and it increased in both the salinity with and without sulfate, and it did not really change that much in the control or the sulfate. Notice that these have different axes. Now, it took me a while to publish this because for a long time, I didn't believe it and I thought it was wrong. I did eventually publish it, and then more recently, we combined our results with results from other lab and field experiments in Delaware and Waccamaw, and then also observations along the salinity gradient in the San Francisco estuary. And it was good to see, here's our studies, you see increasing methane with the salinity. In the San Francisco estuary, they also found methane increasing. Where experiment, they also saw it, but I guess the data that they used here was not where they saw increased methane. So it was good to know that we weren't the only ones that found increasing methane with salinity. Furthermore, we had taken soil cores, and it took a while to get the microbial data. And I know there's a lot going on here, but the experiment we did is here in the alligator, and you can see that microbial communities become less diverse. You have the freshwater versus the oligohaline, so the salt addition. So there's less diversity in the microbial communities, and there's changes in those functional groups as well. We also looked at gene frequencies across these different experiments, and 19 of the genes were correlated with methane fluxes, with gene abundance. Most of those were positive. A couple of those were negative. But again, it was interesting that it was salinity that was affecting the microbial community and the gene expression, and it was not sulfate. We did not see sulfate really change anything in fluxes, nutrients, microbial community, or in gene expression either. So we've also done lots of studies doing surveys across the Albemarle Pamlico Peninsula. So Steve Anderson had worked with in Emily's lab. He came and did a master's with me, and then he went back to Emily's lab. We collected a bunch of different plots and came up with species that tend to be in the forest, species that tend to be in transition, and species that tend to be in marsh, and looking at soil salinity and elevation were the biggest explanatory variables in terms of the community composition and how it changed across this landscape. Steve also did a nice greenhouse experiment where he took different species of trees and added different amounts of salinity, and then looked at the reflectance and calculated different indices, the NDVI, PRI, and found changes in those indices as you would expect. And we'll use that in a little bit later with remote sensing. My PhD student, Melinda Martinez, who's now at the USGS, was really interested in greenhouse gases, and I like this link, right, that the tree is a passage between the earth and the sky. And so we got very interested in what are these trees doing in terms of greenhouse gas emissions. Are they a straw that facilitates the movement of gases between the soil and the atmosphere, or are they a cork in keeping gases in the soil? So Melinda had these flexible chambers and measure emissions from the snags, and she also measured it from the soils. She measured CO2, methane, and nitrous oxide, and we found that the snags are emitting CO2. They're emitting a little bit of methane, a little bit of nitrous oxide. The snags emit about 25% of the soil emissions. So the soil emissions are definitely higher. but they're definitely not zero. So these snags are emitting these greenhouse gases. When Melinda wrote this paper, we talked to our communications office and told her all about the work we had done, and at the end we said, oh, and as a joke, we call these tree farts because I have young kids, and that's how I have to tell them so they get interested in my work. And they put that in the news release, and then the science media loved the idea of these tree farts and put it everywhere. I think Melinda talked to reporters for nonstop for like 2 weeks about these tree farts. Um, we also measured the methane concentration at different heights of the snags and looked at the isotopic composition of the methane, and we found that the methane was getting oxidized as it moved up into the through the snags, from the soil into the snags. So yes, the snags were functioning as straws, but really they're more like filtered straws because they're changing the methane as it gets oxidized. And I also like to use this to tell kind of the general public that these trees, even when they're dead, they're still helping us with fighting climate change. We also took cores from these snags and look at the a microbial community, and the microbial community were mostly methanotrophs. There were not many methanogens in these microbial communities. MJ Carmichael just published this paper recently. She was the one who led that work, and it was nice to find microbial community composition that correlated with the flux data. We care about carbon accumulation, carbon sequestration, right? So Jillian was another grad student. She likes to get to the core of the problem. She collected a bunch of soil cores, and then we used lead and cesium dating to estimate carbon accumulation. And so here I am comparing it to a couple of, Chris Kraft had done the same thing in Georgia. He had found that marshes were a lot better at sequestering carbon than forests. Greg Noe did the same thing in Georgia and South Carolina, and he found the opposite. We also found that the forests sequester more carbon than the marshes. When we're looking at nitrogen, again, Chris Kraft's marshes are doing amazing. Greg Noe's are not doing as well, and ours were pretty much equivalent, right? So it seems that for us, if the change is from a forest to a marsh, if what you care about is carbon or nitrogen, it might not be that big of a change, right? In terms of carbon accumulation. Now, we also looked at where that carbon was, right? And so from Jillian's data, we estimated carbon stocks in the soil. And then from that Lindsay Smart's paper that I mentioned earlier, we looked at carbon in the biomass. And so in some of our sites, most of the carbon is in the biomass. In the forest, less is in the soil, but there are other ones where more of the carbon is in the soil and less in the biomass. In some of our transects, the marshes have more carbon than the forest, and some of our transects, the forest have more carbon than the marsh. And then I borrowed from Matt Kerwin this calculation of replacement time for the carbon in the biomass based on the carbon accumulation rates of the marshes. So in some places, it would take 200 years for those marshes to sequester the amount of carbon in the biomass. And in other sites, it would take 600 years for these marshes to sequester that amount of carbon. Now, if you remember that map I showed you earlier, these marshes do not have 200 to 600 years, right? So it's really important that we figure out ways that we can, protect these forests and protect this carbon, because it's unlikely that these marshes would be able to sequester that carbon in the timeframes that are available. So another thing that we've been working on, and this was Melinda's Martinez work again, she was using NDVI, using the Landsat, and using the change in NDVI over time to map the trajectories for this landscape, again, the Albemarle Pamlico Peninsula. Our hope was to determine early warning signals by looking at things like critical slowing down, increasing autocorrelation, increasing standard deviation. And what we found is that those worked, but it was only in a small percentage of the landscape where those early warning signals popped up. And part of it was that all the trajectories of change were very different. There were places where the landscape was getting greener, right? And those have a positive NDVI trend. And in those places, the standard deviation over time was getting smaller. So we call those stable forests. There were some places where they were getting greener, but the standard deviation was increasing over time. And we found those mostly were places recovering from wildfires. And then places that were getting browner, so the NDVI was decreasing over time. Sometimes that transition was occurring slowly. So there were no really early warning signals, but really it was just happening slowly. And then sometimes we would see an abrupt transition. And in those cases, sometimes we would see critical slowing down before, but other times we were not. But this kind of mapping of the trajectories we think is helpful for management because these areas right here, you might not want to do anything about. These areas right here. You might want to go in and put in water control structures, artificial reefs, maybe even seawalls in those kinds of areas. And then we could map these onto the landscape and put in the trajectories so that people can decide where they want to go in and put in those limited conservation dollars. This area received a large amount of money from the infrastructure bill, and they're deciding now on how to use it. This paper is, I put it here in revision. It's in press. It should be out already, but it should be coming out soon, and I can't wait to share this map with folks out there that are making these decisions. Again, trying to look at larger scales, I've also been using the forest inventory analysis data set. And so as you might be familiar, this is a large, national data set where they have these plots and they go out there and they sample them over time. I used data from the two most recent surveys to look at change. This gives me about a decade of forest change. And I used plots from what we called low elevation counties and then mid elevation. So the low elevations were about 5 meters or less. Mid elevation counties are 30 to 50 meters. What you can see here is that there's a lot of heterogeneity here. If we focus in Louisiana, right, there are areas that have lost a lot of forest right next to areas that have gained forest, right? And this makes sense because there's agricultural abandonment, there's areas that are just regrowing. But if trying to understand the growth and the mortality, there are relationships. So places that had higher rates of sea level rise had lower growth than if you had lower sea level rise rates. And then same with mortality, the number of storms led to increasing mortality. This is also comparing just the low and the mid elevation. So the low elevation counties had lower growth and higher mortality, and that was not driven by the harvest rate because the harvest rate was opposite. We also looked at the change in biomass between live trees, standing dead, and coarse woody debris as well. I guess I'm getting to the end here. One thing I wanted to mention that I've been working on here, in addition to doing the research and trying to do some more outreach, is using citizen science, right? And so we started this project a couple of years ago, the Sentinels of the Sounds, and I've used different platforms. What I'm using now is Chronolog. And we set up these stands where people can come in and put their cell phone and take picture of the same area over time. And then there's instructions there or where they can email those pictures. And then those pictures go to this website where you can go in and see the time lapse. So initially I did it in 5 areas. One that was like really a ghost forest and a couple that were kind of healthy forests with the idea to try to capture that change. One of the sites is Goose Creek State Park. And so the North Carolina State Park system was like, oh, this is great. We need more of these. So we ended up adding like 14 more in different state parks. And now we've added one in a national park as well, trying to capture the change of these ecosystems over time and trying to involve people through this. All right, I'm getting to my 30-minute time limit. I'll just mention here briefly, right? So I started doing all of this work in terms of salinity and ecosystem change because I was looking at wetland restoration, right? And restoration in general is a backwards looking practice, right? We have this idea that we had a forest, we developed it, and through restoration, we're going to bring it back. Unfortunately, we can't really do that anymore, right? Because climate change, sea level rise, more storms, all of these things are changing those ecosystems. And so we need to think better about how do we make decisions on the landscape? What are this landscape going to look like in the future? Because what those targets that we're backwards looking are not really realistic anymore. And so we need to understand those trajectories of change so we make better decisions on these landscapes. And so, okay, some conclusions here. We know that ghost forests are expanding. The drivers are not fully understood. Salinity is important, flooding is important. The combination of those is probably important as well. Salinity can alter gene expression, microbial community composition, ecosystem processes, and the effects might be different depending on where you are, depending on your soil type, depending on your hydrology. Remote sensing of these trajectories can open windows of opportunity and allow managers make better decisions for these landscapes. And with that, I'll stop and I think have plenty of time for questions.